Eva Sapi. I'm a professor at University of New Haven, and I established a research team for Lyme disease about 10 years ago. And to date, I had over 70, 80 students who finished a project, either a thesis project or research project with me in the field of Lyme disease. What we do, we want to kill Borrelia burgdorferi, the Lyme disease bacteria. It's that simple. One of the other plan is to try to see what kind of pathogens you can find in ticks. However, the recent efforts in the last couple of years is mainly focused on understanding why we have hard time to kill Borrelia.
Okay. Your team determined that Borrelia burgdorferi is capable of forming biofilm in vitro. Would you please tell us more?
Yes, a couple of years ago we started to expose Borrelia to different adverse conditions, like, we put some antibiotics in the media, or we changed the pH, or we changed the temperature. And what we started to see that Borrelia started to have this defensive morphology. And one was very interesting. They started to have these big aggregates. And first we didn't really understand what it means. So we started to look at those aggregates, and turned out that a lot of other bacteria can form these aggregates, and it's not just the aggregates, but even has a protective layer on these aggregates, and it's called biofilm. And we learned that if the Borrelia are capable of making this biofilm, actually it becomes very, very resistant to antibiotics. So in a 2011 paper, actually, we studied the antibiotic resistance of these aggregates, and we found it at least 100-fold more than the spirochete. So after we got very interested, to see what can we learn about these aggregates. Maybe if we understand better, maybe we can have some strategy how to eliminate it. So in a 2012 paper, we tried to have some very basic fundamental properties of these aggregates. We found alginate as a protective layer on it. We found that it is a huge morphological rearrangement in these aggregates and forming these structures called the channels, which now we understand the channels, actually the part when aggregates can communicate with the environment, bring the nutrition in and get the waste out. It's very important for the aggregates. So obviously when we published this paper, nobody believed us. They said it's not such a thing, there's only very few bacteria can cause biofilm. So one of the major criticism was that they never seen this in animal models. So when they introduced Borrelia into the mouse, for example, they never seen anything like that. So I searched the literature if it's true or not, and actually I found a couple of very, very promising pictures from some old experiments when actually indeed they look like they've seen some aggregates. And it's an Austrian study showed some skin condition, which is Borrelia skin condition. They also see something very, very similar. So I talked to the Austrian researchers, and I asked them if they can send me some samples from the studies, and actually they did send me some samples. And we just finished, really just a few weeks ago, a paper when it's describing these aggregates right in the skin conditions, so in vivo, not just in vitro, not just in a lab, but in the tissue, in the human tissue, and we provided a set of evidences that this is indeed exists. So right now the paper is ready to go, and right now we're just waiting for one more PCR data, and we would like to send it within a couple of weeks. So hopefully that paper gonna finally prove that Borrelia biofilm indeed exists, and we need to really take it very seriously because this is an extremely antibiotic resistant form. The other study which is right now is underway is a mouse study. Remember, they told me that they couldn't see these aggregates in mice. And so I was very curious why not, because we have seen it obviously in the skin tissues. So one of my colleagues, Jennifer Miller, she actually was performing a mouse study for another study, not for us, but she said she can send me some leftover tissues. So she did, and she sent me heart, brain, liver, obviously skin, and some joint tissues. And we started to screen it for these aggregates. And I have to be honest, the first screening, we didn't see anything. And I said, okay, all right, so they were right. It's nothing there. And suddenly we reached the heart. We started to look at the heart, and it just showed up like a Christmas tree. The bottom of the heart, which is the low oxygen, I guess probably the lowest oxygen condition, showed a lot of aggregates, huge aggregates. This is another study we tried to finish. And of course, I'm sure you're going to ask the question, how about the brain? Because everybody, Lyme, yes, brain. And we just started to screen the brain, and indeed we found the aggregates. So those studies are still underway. We need to finish some PCR data. We need to finish some additional staining data, but those studies hopefully will be finished, I would say, by October, November. So this year we were able to send out a couple of papers showing that those aggregates, those biofilms, indeed are in the tissues.
So I guess you have already answered a little bit to this question, but what's the organization and composition of Borrelia burgdorferi biofilm, and are there only one or different biofilm models?
Okay, I didn't hear the question. Could you repeat one more time?
What's the organization and composition of Borrelia burgdorferi biofilm, and are there only one or different biofilm models?
Very, very good question. Yes, we strongly believe it is more than just small model. We try to see, we have a couple markers for biofilm. In a PLOS paper, we identified the alginate as a marker, but recently we found other markers which was proven from other biofilms. Borrelia looks like expressing some of those markers. I have one project which is try to sort of describe different markers. And what we have seen is those markers are not always expressed. So definitely certain conditions favors certain markers. The other thing we've seen that anytime we studying this biofilm is this huge heterogeneity in the experiment. So sometimes we see, for example, I'll give you an example. We treat them with doxycycline. And I have this one figure I always show to everybody. We treated some in vitro biofilms, so in a lab, for 3 weeks with doxycycline. And after the 3 weeks, we have seen dead biofilms and we have seen live biofilms. So some biofilms were able to survive, but some biofilms were dead. So that already shows that there are huge heterogeneity in the biofilm. So it definitely has to be studied further, because it may be, this is one of the survival mechanism for Borrelia. If we would have just one kind of biofilm, we could figure out and we can just eliminate it, yes? But if we have different types of biofilm, we really need to understand what are the differences and how they will going to respond to the antimicrobials.
Okay. And what would be the role of biofilm in Borrelia burgdorferi life cycle and in Lyme disease?
That's obviously the big question. So okay, so let's see that we proven that the biofilm exists, is in the tissue, in a different tissue. So does it affect the treatment? And again, this is one of the things we're trying to answer. There's a very famous case, Dr. Ligner case from New York, when he treated this woman for a long time with antibiotics, and every time he treated her, she was doing fine. Unfortunately, because some insurance issues, they couldn't treat her anymore. I guess she couldn't afford the treatment anymore, and actually she developed a very huge inflammation. The inflammation they couldn't control, and actually she died. So it's a very sad story. The only good news that we have the tissue available. It is in Columbia right now, Columbia University, and we already had chance to look at some of the tissues from this case, and we we seen a biofilm. So the question was, is biofilm caused of those huge inflammation? This biofilm was the reservoir for Borrelia. So every time she got treatment, the biofilm maybe got some treatment but wasn't eliminated. So the minute she didn't have the treatment, obviously the whole thing resurfaced. Again, what was this huge inflammation? Is biofilm causing inflammation? I mean, other bacteria, they showed that indeed biofilm can cause very significant inflammation. Now, Lyme disease. I know we have lots of different symptoms, but one of the main symptoms is the arthritic symptom, the neurolyme, all those conditions which can easily explained by the inflammation. Because inflammation is practically your immune system trying to eliminate a bacteria, a pathogen, for example. I talked to a lot of people saying that, it's an autoimmune condition. I personally don't believe in autoimmune conditions. I think autoimmune condition is nothing else, just an infection which wasn't treated properly.
And what is the impact of Borrelia burgdorferi biofilm and antibiotic tolerance? And what impact do you think your research will have on the treatment of Lyme disease?
Yes, obviously the big question why this biofilm is so resistant to antibiotics. We have seen lots of protective layers. It's not just one layer. It looks like it's several layers is protecting the biofilm. However, we have channels, and those obviously, if you protect your own structure, you have to have ways to bring the nutrition in. So those channels can provide this base, but the question is, can we use the channels for bringing antimicrobials in and destroy the colony from inside? And this is the approach we tried. About 2 years ago, we tried to mimic a Nature paper when they showed that to deal with biofilm, you have to keep those channels open. So how you can keep the channel open? Obviously with food. So these researchers, they added some sugar in the media, different kind of sugar, and at the same time they added the antimicrobial. So the idea was keep the channel open with sugar and sort of sneak in the antimicrobial. This is the Trojan horse, the Greek Trojan horse idea. And actually we have tried it, and we couldn't really repeat the data in our system until we try something called stevia. Now, stevia is a sweetener. Now it's approved even in the US because it has very safety profile. It's from Japan. It's a Japanese leaf. And it obviously, we use it for sweetening our tea or coffee. So when we were doing this sugar experiment, stevia was just one of them because we had the whole set of sugar, whatever we could find. And very surprisingly, we found stevia worked very well itself, not just waking up those those cells inside and opening the channels, but also as an antimicrobial. So we researched the literature, and we found that stevia actually used as an antimicrobial before. So right now, just a couple months, we compared stevia with some promising new antimicrobials, and we were very surprised to see that stevia worked very, very well. So again, we're thinking the idea is behind it that stevia is a sugar, so sort of opening up the channels, and stevia also antimicrobial, so it has a two purpose. So we were wondering if we could have similar compound which has sort of a food component to it. So the Borrelia thinks it's eating right now, yes? But at the same time, we can bring in the antimicrobial effect, maybe we're able to eliminate this form as well. But again, as I mentioned, there is a huge heterogeneity, so who knows, maybe few clones still gonna survive. It's difficult to say at this point.
And are there any alternative strategies to treat Borrelia burgdorferi biofilm?
I mentioned some herbal approach. We screened over 100 different herbs, and agent, which we know people are using. I'm sure you heard about the bee venom. A lot of people are using bee venoms. We actually have one project finishing up with one of my students. Looks promising. I don't know how easily can be administered bee venom. Some people I heard they just let them stung by the bee. So I seriously don't know how can we use that. But we think from our screening all those alternative agents, definitely nature provided answers for eliminating Borrelia, and I think we need to explore a little bit more. Antimicrobials, we have done a lot of antimicrobials antibiotic screening, and they do work, but somehow they also creating a lot of resistant clones. So we feel that we need to address this whole antibiotic resistance with different approaches. Can we maybe combine antibiotics with maybe some herbal component? Maybe. One of the interesting idea also we working, and this came from a doctor who was using lactoferrin combined with some antibiotics, and I didn't really understand first. But after now, when we started to try it in a lab, we found it's very effective. And I said, what could be lactoferrin? I mean, it's milk practically effective. And turned out lactoferrin can chelate out iron, which it's not a big deal because Borrelia doesn't use iron, but also manganese. Now, manganese, however, is important for Borrelia. So we have this idea that maybe manganese is the one which needs to be maybe eliminated, and maybe lactoferrin can do that. So again, some people combining alternative therapies with some standard therapies and getting success. Some people are just using regular therapies, and some people are just using alternative. One of the things we screen the Cardon protocol, we screen every single herbs in the Cardon protocol, and some of the herbs were very, very effective against Borrelia. So again, what is the best approach for the treatment? Should we use heavy antibiotics, or should we look into the nature and see what nature provided?
Now, one more general question. How could we manage Lyme disease infection?
I mean, obviously, the best management is to eliminate Borrelia, and as you see, there are a lot of challenges here. This spirochete on this planet now for I don't know how many billion years, so obviously figured out how to escape not just from the different adverse environmental conditions, but from our immune system. Very smart bacteria. So number one, we have to learn how to eliminate this bacteria. Number two, I heard a lot of people getting better, just healthier lifestyle, eating healthier, eliminate the sugar, and try not to just sit on a couch, but move the body. I heard people getting better, even just increasing the temperature. They're taking these very hot baths, and Borrelia doesn't like heat, that's for sure. So that might be one of the things we can do. Some of them taking infrared sauna, which is a very interesting approach because it was some suggestion that certain frequency actually is not good for Borrelia, and infrared sauna obviously provides certain frequencies, not just the heat. Other people got better just to do detoxification. Some people believe that people who has chronic Lyme, they have some defect in detox pathways. So certain nutrition can help to overcome those defects. Right now, I just talked to somebody who said that there are genetic panels they try to develop, to see if a Lyme patient, the one who is not recovering from regular treatment, maybe had this genetic component to it, and maybe that's really the key to really get them better, to support those, let's say, the liver detox pathway, these two major ones, so maybe add nutrition to support both pathways. Probably, I had Lyme, and I was working with this natural doctor, and he believed it very strongly that the detoxification should be very, very important. So he was giving me a nutrition supplement which support liver pathway 1 and 2, and I have to say I really, really felt better right away. So again, it's more than just killing Borrelia. I think that body needs to be supported. Can we kill Borrelia in the body 100%? Probably not, but don't forget we have immune system. If the immune system is healthy and working, obviously can finish the job for us.
So as a last question, I would like to know if you want to tell us a little bit more about your story with the Lyme disease.
I think my story is not that unusual. When I started to have symptoms, my Lyme test was negative. Even they suspected it was Lyme. The only reason that finally they agreed that it might be Lyme because I had the MRI, I had neuro-Lyme, so I had the MRI and they saw the lesions, and the lesions were very characteristic for Lyme disease. But even that, they were very hesitant to give me any antimicrobial. So at this point, I actually used alternative approach because I didn't feel good. And it's not because I was loving the alternative approach. I had not much choice there. And recovering mainly using alternative approach, I see that is definitely more into it than just giving the patient antimicrobials. How long? Everybody asked me how long did it take. It took me at least 2 years. Depends on where you count, yes. And it was some relapses during these 2 years, but I started to see improvement after a few months. So I knew I'm on the right track. I just keep doing. My diet was perfect, as perfect as could be. I did do light exercise. I did do the far infrared sauna. And you know what? The most important thing, I strongly believed I'm able to recover from this. I didn't believe that I will have Lyme disease forever. So I think this number one is sometimes patients really giving up, and I think it's very important to tell them that you can recover from Lyme disease. Just you have to know your body and work with your doctor and let the doctor know if the treatment is not working.